Hello, everyone. So today we're going to talk chi-squared and genetics. So one of our science practices is being able to use statistics to analyze results. Today we're going to practice using chi-squared to analyze results from a genetic test to see if we get variation from our predicted outcomes. So you may recall we did chi-squared back at the beginning of the year as part of our unit 0. And so today now we're going to apply that, let's see what time is it? Okay, apply that to genetics instead of just experimental results like we did with, it was M&Ms and some pill bugs back at the beginning of the year. So it's important to remember chi-square is a statistical test that's used to determine whether or not the results of a scientific experiment can be accepted. So whether or not they were just random chance or if they were actually due to a variation. So when it comes to genetics, we talked about our Punnett squares and how we have Mendelian genetics and things that are dominant versus recessive. So we're going to be able to use chi-square to see if things follow that Mendelian pattern that we expect, or if there's some sort of non-Mendelian thing going on, like incomplete dominance or linked genes. So with chi-squared, it helps if you can remember some of our common Mendelian phenotypic ratios, so what things look like, phenotypic. With monohybrids, some typical ratios that show up. So we have a 3-to-1 ratio. That's when both parents are heterozygous. There's also a 1-to-1 ratio. This is the reduced form, or 2-to-2. If we were to cross a parent that's heterozygous with one that's homozygous recessive, like in a test cross. Our dihybrid typical ratios, if both parents are completely heterozygous, like this one down here with our P's, then we have a 9-3-3-1 ratio. If we have a homozygous recessive parent crossed with a heterozygous, then we get that 1-to-1-to-1 ratio. So here's an example using some fruit flies. And we had 2 flies that were both red eyes, were crossed, and their children, now flies have way more than 4 children, have 73 that have red eyes and 27 that have vermilion or a white eye, a pale eye. So our probable Mendelian ratio, we've got a big difference, right? They're not close to being 1-to-1 or equal. So we can say 3-to-1 with the probable parent genotypes of being heterozygous. Now what we're going to use chi-squared to determine is if 73-to-27 is close enough to what would be a perfect 3-to-1 ratio of 75-to-25. Since we're doing this out of 100 possible offspring. And so that's where chi-squared comes in. Is that really a 3-to-1 ratio? And it just looks a little weird because of chance, or is there something else going on? So here in our chart, and our null hypothesis, anytime we're using chi-squared with genetics, is that what we observed does not vary from what our expected ratio is. So we're testing that our observed ratio does not vary from the expected ratio. So here with our red eye versus our white eye, which is vermilion eyes, we observed, it told us in the problem, 73 and 27. As far as our expected goes, out of the 100 offspring, so I multiplied the 100 offspring by the fraction that we get from our ratio. So our 3-to-1 ratio, that means that we have 3 out of 4 possible outcomes. So 100 times 3 out of 4 gave me that 75. That's how I got my expected. I find that most times students struggle with figuring out the expected values. For the vermilion eyes, I did 100 times 1 out of 4. Because we look back here at our square, 4 possible outcomes. We had 3 with red eyes and 1 with the white or the vermilion. Then I can do my chi-squared applications. So I do my observed minus expected. I square that. And then I take the number and I divide it by my expected. I divide it by 75. Here, 27 minus 25 is 2. I square it, and then I can divide by 25. To get a chi-squared value of 0.213. So now we have our chi-squared value. Now we can look at our chi-squared table. This is something that's always given to you on your AP Biology formula chart to determine if we're going to reject or accept our hypothesis. Again, that there is no variation, no difference between our observed ratio and our expected ratio. Things to remember, we're always going to use our p-value of 0.05. Degrees freedom is going to be n minus 1, where n is the number of classes or groups. In this case, it's phenotypes. So we had 2 phenotypes. We take away 1. We end up with a p-value of 1. So we're going to be over here in this table, this column of our table. And then we have to compare our chi-squared value. If it's less than the critical value, we can accept. We can say that null hypothesis is most likely correct, or we can reject. So in this case, as far as our conclusion goes, we can say that we accept the ratio as being 3:1 because our chi-squared value, which was 0.213, is less than the critical value, 3.84. One more practice problem. This time around, we're going to look at a dihybrid cross using some corn. So corn's got way more offspring than just 16, looking at all the kernels on a corn seed. And it gives us some observed results. It always follows a pattern of both dominant traits and dominant recessive, recessive dominant, and recessive recessive. So we have 914 yellow round kernels, 286 yellow wrinkled kernels, 297 purple round kernels, and 103 purple wrinkled kernels, with a total number of offspring of 1,600. So for our expected Mendelian ratio, so these aren't even, they're not 1, 1, 1, 1, 1. So we can most likely say that's probably a 9,331. You can even test it out if you were to take this 103, multiply it by 9, you're not all that far off from 914. Not perfect, but there's variation. What we're trying to figure out if that variation is due to chance or due to something else. So as far as our expected values go, so here's the math I did. I took our total number of offspring, multiplied it by 9 out of 16. If I go back here, this ratio we originally had 16 squares to it. For yellow wrinkled, I did the number of offspring times 3 out of 16. I did that for also the purple round. Then when both of them were recessive, 1,600 times 1 over 16, give me 100. So now to compute our chi-squared value, so we can use our chart again. And so comparing our observed to our expected, and here we have a chi-squared of 0.991. And now we can analyze that for an answer. So there's our chi-squared value. Our degrees of freedom, this time we had 4 groups. Take away 1, that leaves us with 3 degrees of freedom. So on our chart, our critical value now is 7.82. So there's our critical value. Is our chi-squared less than the critical value? That's going to be yes. So we can now accept the 9,331 ratio.